Hello everyone, I'm Irsyad Ilham Basuki from Group 2. So for this video, my group and I are going to be presenting about the basic chromatography, specifically for the CHM256 video presentation. Without further ado, let's start our presentation. But before that, let me introduce you to our team members that contribute a lot to this presentation. It consists of me myself, Irsyad Ilham, Fariz Ikhwan, Azim, Sulaiman Nawi, as well as Wan Amir. So, what is shown in the slide is the table of content, basically what we are going to be presenting throughout the video. So, one might actually ask this question, what is actually chromatography? But no worries, let me introduce you to the world of chromatography. Chromatography is actually a technique in which the components are separated based on the differences in rates, at which they are carried through a stationary phase by a mobile phase. Chromatography analyzes and separates mixtures of compound in different types such as gas, liquid, and paper. For your information too, it is commonly used for separation, identification, as well as determination of chemical components in a complex mixture.
So, Assalamualaikum warahmatullahi wabarakatuh. My name is Mohd Fariz bin Asriya. Today I will continue this presentation with theory of chromatography. So first of all, chromatography separate and identify mixture component by passing them through a stationary and mobile phase. And different component interact differently with stationary phase and move at different rates, resulting in separation. And this make it possible to analyze and identify individual component. Adsorption, partition, ion exchange, and size exclusion are four different forms of chromatography that each have unique application and advantage.
My name is Wan Amir, and I will be presenting the principle of chromatography. Samples usually is a mixture of many components. The component of a sample with unknown compounds must be separated prior to identification by using other analytical methods. The intermolecular attraction of each component is also different. Some components will be more attracted or absorbed towards stationary phase, while some will attract towards mobile phase. And from here, the component will distribute themselves.
Next, what is stationary phase? The stationary phase in chromatography is a fixed material through which the mixture is passed. Other than that, it provides a surface for compound to interact with and play a crucial role in the separation process. And different type of chromatography use different type of stationary phase. And the next slide, what is mobile phase? Mobile phase in chromatography is a fluid that carries mixture through stationary phase. Can be gas or liquid depending on type of chromatography. Properties of mobile phase can affect separation process. And the last one is different chromatography use different type of mobile phase. And that's all from me. Thank you.
Hi, my name is Azim bin Jaidon. Today I will present about the classification of chromatography method. There are two types, which is planar chromatography and column chromatography. Next is classification of planar chromatography. In the type of chromatography, the stationary phase is put on a flat plate or in the pores of a paper. The mobile phase moves through capillary action. The example of planar chromatography is paper chromatography and thin layer chromatography, which is TLC. Next is column chromatography. In the type of chromatography, the stationary phase is held in a narrow tube. The mobile phase is forced through the tube under pressure or by gravity. The example of column chromatography is gas chromatography and supercritical fluid chromatography.
Next, I'm gonna continue the video presentation with my next part, which is types of separation. For your information, types of separation is divided into two different parts, which are partition chromatography and absorption chromatography. So let's start with the partition chromatography first. Partition chromatography is a distribution that dissolves the component between two immiscible liquid phases. It is a type of separation where components partition themselves based on their relative solubility. In a way, solid equilibrates between both liquid mobile phase as well as liquid stationary phase. Next is the absorption chromatography. Absorption chromatography is a separation process where the stationary phase is a solid, on which the sample components are adsorbed. In this particular process, the mixture's component specifically absorb on the surface of a finely divided solid stationary phase. The mixture's component stick to its surface with different degrees of strength and then separate as the carrying process occurs.
Hello, my name is Sulaiman Nawi, and I will be presenting the methodology of TLC and also the results of the experiment. So firstly, for the methodology, the first step is to prepare and activate the TLC plate. And after that, we can spot the TLC plate with the sample. Then for the third step, we'll be developing the TLC plate. And afterwards, for step 4, we'll measure the RF values. And after that, we can start to visualize the steps and swaps. And lastly, for step 6, we dry the plate. And moving on, we have the results of the experiment. So we can see all the types of pigments and the colors that were obtained from the pigment. So for B carotene, we get the color yellow. For chlorophyll A, we get a blue-green type of color. And for chlorophyll B, we get a green. Lutein, we get a yellow. And neoxanthine, we get also yellow. So that's it for my part. Thank you for listening. I'll be passing it on to my other group members.
Next is application of planar chromatography. The first one is separation of components in a mixture, such as ink and dye components. The second one is color pigments dispersion in plants. Then, pesticide or insecticide identification in food. And the last one is analyze the dye composition of fibers in forensics. Next is application of column chromatography. The first one is pharmaceutical, which are antibiotics and steroids. The second one is food industry, which are antioxidants and artificial sweetener. Next is forensic chemistry, which are drugs and poison. And the last one in industrial chemicals, which are surfactants and condensate aromatics.
In conclusion, basic chromatography is a versatile and analytical technique that separates and analyzes mixture of substances based on their differential interaction with mobile phase and a stationary phase. It has proven to be an invaluable tool in various fields such as chemistry, biochemistry, forensic, and environmental science. By exploiting the principle of adsorption, partitioning, or affinity, chromatography allows for the identification and quantification of individual components within a mixture. Its wide range of applications, simplicity, and high resolution make it an essential method for scientists and researchers worldwide. With ongoing advancement and innovation, chromatography continues to evolve, offering improved accuracy, sensitivity, and efficiency for the analysis of complex mixtures in diverse scientific disciplines. And that's all from us. Thank you for taking your time to watch this video.